Next up is Evan Newell. He is a postdoc with Mark Davis and is one of the small group of non-Nolan Saitov pioneers. So he's going to be talking to you today about some ways to analyze complex data such as Saitov data.
Thanks a lot. And then I'm also going to try to build on this little conversation about cell subset diversity from a, kind of a simple-minded perspective. And so I talked yesterday about the data that we were generating, looking mostly at CD8 positive T cells. And so I'm just going to sort of continue on that and then use it as a demonstration for ways that I've been messing around with this data, basically. And I'm sure those of you who have like more mathematical aptitude have been chatting with people in Gary's lab who have done a lot more. So I'll just give you some of the ways that I've been messing around with it. And there's obviously a lot more that I can't cover that they've already been doing as well. And so there are just many avenues here of where you can go. And so so my goal then is just to inspire new ways of thinking about this and looking at this data, because we're getting more dimensions than we can comprehend. And so new ways of trying to look at it will be useful. And then I'm going to build on the data that I introduced yesterday with the CD8 positive T cell Saitov experiments I was doing. And most everything I've been doing is in MATLAB, just because I have sort of dedicated some time to learn how to use that. But this could be done in any platform that can deal with matrices of numbers. And as I was saying, I'll just defer to many others around here and that you probably know with broader knowledge on other sophisticated approaches. And then one of those is SPADE, which is very nicely implemented and broadly applicable. And it's being used a lot, as you've seen, and definitely something that we should continue to use as well. So again, just to illustrate this problem is that now we're up to about 40-ish channels. And so each cell we get 40 parameters of information. And how can we comprehend this 40-dimensional space? And of course, I don't need to introduce how this is happening. You've seen that many times. I'll show you again my panel that I've been using for this analysis. So it's 36 parameters of T cell phenotyping. Some of them are just to identify cells and some internal controls. Most of them are surface markers and tetramers. What I normally do is I gate on whether or not they're tetramer positive, so the antigen specificity first, and then just look at the surface markers of those cells, as well as the intracellular cytokines and other functional markers, and so the object is to try to deal with that. And so again, I showed this yesterday too, how we get these data. We just so again, gating on the cell subsets. In this case, just very exclusively live CD8 positive T cells, and then also the antigen-specific ones when it's applicable. And so, again, this challenge is, okay, we've got all these, we can look at the data and make sure that we're happy with the quality of the staining for different markers, the surface markers here, and you can already see that there are certain subsets you can gate these cells into, and this is one of the classical ones, the CD27 versus CD45 array, which distinguishes basically 4 different types of CD8 positive T cells. These would be the naive cells, and then when they lose CD45 array, they become memory cells, and these would be effector cells that are also lost CD27. And then the latest stage effector cells regain CD45 array expression. And so the question is, how do we fit this paradigm in with all the other markers, and how accurate is it? And combining that with all the functional capacities of the cells. And it's nice with the Saitov that we have pretty good signal when it comes to looking at a sledgehammer stimulation induction of cytokines, and this is, in this case, a probe of functional capacity because we're just hitting them really hard. And so, in general, this is sort of what I want to go over in this talk: is that we put cells into the Saitov, and then we get an FCS file out. And I showed you some FlowJo analysis. You could just as easily do this in Cytobank, And what's nice about Cytobank, as you heard, is that they're incorporating these new ways of looking at data as well. I just happen to be a little bit more familiar with FlowJo at the moment. And then, like as Aaron went over, and also this is again being incorporated into Cytobank as well, the SPADE approach. So I'm not going to talk more about that, but you can also do the SPADE approach using FlowJo gated samples if you export a text file and then put it into the system that Aaron and colleagues developed here. But what I've been doing is I take export a text file of those gated cell events that I showed you, and then put them into MATLAB. And from there, I'll show you some simple-minded approaches of dealing with these matrices of numbers. So there's sort of borrowing from the bioinformatics and gene expression analysis tools that MATLAB has. You can look at cells that way as well. We're also sort of extended this Boolean gating scheme that I'll show you to accommodate more parameters. And then I'll talk about a classic way of analyzing data, principal component analysis, and how I think it can be used to give you some sort of intuitive feel for what kind of populations you have and how they might segregate. And these are going to give you comparable information, but it's just a different way of looking at it. And then I can also put FCS files from this. I can export back FCS files that have these new parameters and plot them versus other things in FlowJo. And I'll show some of that, I think. So just in general, flow cytometry data is simply a matrix of data where each row is a different event. So at each row is a different cell, and then each column is a different parameter. And so when you export it as a text file, you have this simple matrix of cell number, what time the cell came in, the length of the cell, in this case for CyTOF is another parameter that gets stored, and then you have your endless number of parameters here. So you have this 40-something dimensional matrix here, or columns of data. And so I normally just work with those numbers and then the header that has the names of each of the parameters in a text file that can be produced in FlowJo or Cytobank as well. Can export text files that then you can open up in any kind of program like Excel even, or MATLAB as I'm doing, or R, depending on what you're more familiar with. And so, this is sort of an overview of the possible things you can do, or sort of some of the possible things you could do with data like this. The most obvious is supervised gating, so you know what you're looking for, and you gate on these populations and look and see how percentages of cells vary between your groups. And this isn't going to go away. It's definitely a good way to start and try to get your head around what's happening. I'll show you some. And then there's clustering algorithms, which is most nicely implemented with SPADE. But you can also use a little simpler approach just to get an overview of everything. And I'll show you how I've been using this. It's certainly not a local favorite. My boss hates this. But it still gives me a good way of looking at it, I think. It's hard, though, to compare samples, and I'll show you why that might be the case. Then I'll show you some crude, correlation or covariance analysis that you can do. And Eli talked about this a little bit yesterday. Once you get these matrices, you can do these other forms of more sophisticated analysis, like these path analyses or these, I don't know what they're called. So, but from my perspective, again, you can just look at this to give you some sort of intuitive interpretations of what's going on in your data, like what things covary or what things correlate with each other, and does that make sense? And so that's sort of a way of just getting an idea of what's going on. And then this Boolean gating thing, this is I'll show you that this has been a classical way of analyzing T-cell functional capacities. But as it goes up in the number of functions, it gets more complicated. So we wrote a simple script to do this, and also to present it in an incomprehensible way, but anyway. And then the principal component analysis. Of course, there are pros and cons here, and I think there is a pipeline of modern alternatives that are going to make this even better that I think, again, some of the Nolan Lab people are working on. And then after that, it's up to you to come up with better ways of thinking about this. So, start with the correlation analysis, and this is one that I probably get the least information from, but I think, mostly because it's hard to interpret when you're looking for correlations between different markers within a heterogeneous population. It's confounded by the fact that you've got all different cells in there. Nonetheless, you can sort of get an idea of what's going on. And so this matrix, so what this is so this is unstimulated cells, again, just total live CD8 positive T cells. And then this is a symmetrical thing here. So it's actually comparing each of these markers twice. So this marker, so each of these is a different parameter that was measured. And then the color represents the correlation coefficient of that comparison. So every possible comparison between each of these markers. And it's actually really hard to read. But things that should correlate, surface markers that should correlate in here do. So like, CD62L and CCR7 are basically measuring kind of redundant markers on CD8 positive T cells. They're both on central memory and naive cells. And so those are things that are going to have a red mark and be correlated with each other. And then there are other things that are known to not correlate with each other because they're expressed on different subsets of cells. And then after we stimulate the cells, we can see how a lot of the cytokines start to become correlated with each other because mostly memory cells make different cytokines, and those cytokines then become more highly correlated with each other because they're only expressed on memory cells. So, and then I think, this analysis might be more useful if I were to go and gate on particular subsets and then look within that at a subset of markers that I was interested in and see how things correlate. And I put the names of the functions that you can use in MATLAB if you happen to be versed at MATLAB. It's kind of fun to mess around with. But all I use for these is the core coef function and then this clustergram function to cluster the markers together that are similar in these plots. And then the next thing is this clustering analysis. And so again, taking advantage of infrastructure from gene expression analysis packages, you can get an overview of your data. And you'll see, so what we have here is each row here is a single cell. So I've got, I don't remember, a few thousand cells here. And then each column is a different marker that the cells express. And you'll see certain things like CD3 and CD8 are highly expressed on every single cell because I gated on them. And then you see different clusters of cells that represent cells that express similar amounts of each of these things. So it's sort of like a hierarchical clustergram that you have with SPADE, except it does not as sophisticated in terms of how it's created, and then you see all the information for each cluster of cells as you go down here. And after stimulation, you can see that a lot more things light up, and then you can see how things kind of cluster. And you can try to interpret what they mean and how homogeneous different subsets are. So for instance, you can look at these markers and figure out that these probably represent naive cells here because they're not making as many cytokines and they express CD45 array and CD27 and things like that. But then it gets confusing once you get into this heterogeneity and what do you do with that. And also the problem with the way this is set up is it doesn't have this mechanism for clustering multiple samples together and then showing them all separately the way SPADE does, where you can sort of overlay multiple samples. And so every sample that you do, you get a completely different output. And so you have to kind of look through and reinterpret it each time. And so it's not good for really making any statements about the data, but to me it's an interesting way of looking at also sort of the quality and what kind of diversity you have within your data. So I'll leave it at that. So this Boolean gating analysis of T-cell multifunctionality has been around for a while. I think one of the more famous applications of this is looking at the polyfunctionality of T-cells that are specific for HIV in people that are progressors versus non-progressors. So actually, Michael Betts did a sort of landmark study where they compared non-progressors and progressors for HIV, and the people with non-progression, the CD8 positive T cells that were specific for HIV were producing more different functions. So they produced multiple cytokines and were able to degranulate at the same time. And so they were using Boolean gating to represent this. So these are all the possible combinations of 3 different cytokines, and then you can see the percentages of cells for each. And then they also sort of sum up different numbers of cytokines to make these pie charts showing like this percentage of cells are making just 1 function, and then these are making just 2 functions, et cetera. But then, of course, it gets confusing when you add more functions. Here are 5 functions, and you've got however many possible combinations of those. And, of course, the same cells are represented in a completely different way here, because you look at more functions, you get more cells making more functions as well. And so then we sort of took this a level further. So now we're looking at 9 functions and saying, okay, I could draw a giant 512 column bar graph here, but it got pretty incomprehensible. So we just put this into a 2-dimensional matrix. And of course, the problem with this is that the order of these is arbitrary, and it's arbitrary here too. So depending on what order you put these cytokines in, you're going to get a completely different pattern. But if you're consistent, then you can sort of compare patterns and also look at the percentage of the number of different types of cells that you can observe here. And so, again, a pretty simple script to look for positive versus negative events for each marker. And then I use this percentile function to train it based on the unstimulated cells. And then you make your vector that has all these different functional percentages, and then I just reshape that into a matrix and plot it with a heat map function. And so this is total CD8 positive T cell stimulated. But then when you look at cells specific for different epitopes, you get different patterns. And so we think that maybe this way of looking at the patterns of polyfunctionality could be useful, because it will say, okay, let's say one pattern is associated with one type of T cell, another pattern with another, and maybe in disease you'd see peculiarities associated with whatever it is you're looking for. So it's just another way of trying to visualizing diversity here and taking advantage of the multi-dimensionality of things. Working with Bill O'Gorman in the lab, he's looking at monocytes stimulated with different things. And I think, and here again, he's looking at a whole bunch of different markers and trying to compare these, in this case, just these 2 stimuli. And you can see that this representation here, a little bit simpler, just 6 cytokines. You can see that the pattern that is produced between these 2 stimuli on the same cells is very different. And so using this as a way to sort of get an overview of all these 6 cytokines is a little bit easier than looking at a whole bunch of plots like this. And so he's also adding in more cytokines to look at the diversity of monocytes this way. So principal component analysis, even though it had been alluded to earlier here, and also it's been around for 100 years, and based on sort of classic linear algebra, it's still very hard to explain, and I probably don't understand exactly the details of the math behind it. But what this does, what it is a method to reduce the number of dimensions that you're looking at. So we have this 25 dimensions left, as that I was looking at in these CD8 positive T cells, and I want to reduce that dimensionality down to something that I can visualize. And so this is one way of doing that. And what it does is it transfers. So because some of these parameters are correlated with each other, it takes those correlated variables and sort of creates a new set of uncorrelated variables. So each new set of parameters, these principal components, are describing independent portions of the information. So you don't waste your time looking at multiple things that are showing the same thing. And then it also puts another thing about it is that it sort of rotates this multidimensional space into the most informative view. So it stacks more of the information in the first few parameters. So the very first parameter has the maximal variation explained by this linear transformation, and the second gets the second most of uncorrelated data. So by the time you've gone through a few dimensions, you've explained most of your variation within the data. And then also, it's important to note, like the other analysis methods, it's unsupervised. But of course, biased by the number of by the markers we choose to put in there, because you put in different markers, you're going to get completely different information, obviously. I put this in here for you to read. It's another way of thinking about it. Again, let's say you have these two correlated parameters, and you want to explain the variation of both of them in just one parameter. You would just sort of fit this and create this new sort of axis that goes that way, that explains both of these in one shot. And this is what happens when you throw the CD8 positive T cells into this, and you can use this. You can get some information about how well it's working. So in this case, these are plots of the percentage of variation explained by each of the principal components. So there are 25 principal components, starting with 25 parameters. But within the first three, the first one explains about 30% of the variation within the sample. And then this shows the cumulative information explained, and by 3, you're up over 50%. So we're getting most of the information right there. And then you also can look at how these parameters are produced. So these are the loadings. Each parameter is a sum of all of these things for each cell. So you're multiplying this vector by your matrix of for each cell, and you get the new principal component. And so, like I said, this is unsupervised. But what the interpretation of this, as you'll see, is that component 1 explains the difference between naive and memory cells. Component 2 appears to explain differences between early and late stage memory cell differentiation. And then component 3 is a little bit mysterious, but it looks to explain diversity within the central memory compartment. And that this is just an interpretation of what we see. And then again, this is based on this prin-comp function, which is just the principal component analysis function in PyMol. And then what I've done in MATLAB, and then what I've done is used MATLAB. I wrote a script to export a PDB file, just because I happen to be familiar with the way PyMol works. It's just a 3D visualization program that reads these PDB files. And so that's what I'm showing here, is a PyMol graph of this data. And so each dot here, the gray dots are every single cell in the CD8 positive T cells. And this pattern that it creates is unsupervised, so it's whatever the principal component analysis created. And then what I did was I used classical definitions, stringent ones, of each of the CD8 positive T cell subsets, and I colored those dots accordingly. So these green cells are naive cells, and then the yellow cells are central memory cells. The blue cells were thought of as effector memory cells, and then the red cells are these late stage or short-lived effector memory T cells. And then you can see that it kind of has this pattern that also has what looks like a bifurcation from the central memory compartment. So behind there's another set of effector cells that we think may represent another branch of CD8 positive T cell differentiation. And I'll show you a little bit more about that. So the other thing about this is that I did the same analysis on 6 different donor CD8 positive T cells, and they all have they have a similar pattern, and it's most obvious when you look at it. And this is probably the most different one, where it's a little bit scrunched. But when you look at it in 3D, you have the same sort of pattern. You have this sort of folded Y shape, where the naive cells here in red are a more uniform blob of cells, and then you have this spectrum of memory cells. So it shows that there's a continuum of memory cells by this analysis, and then it looks like a bifurcation. And so in these plots, they're also colored by the IL-2 expression induced by PMA and ionomycin. And then, so it makes sense that the naive cells would produce less IL-2. And then the central memory cells here are producing the most IL-2. And then as they differentiate in these two ways, or presumably differentiate in these two different ways, they lose expression of IL-2 on this pathway and then on this other pathway, if that's indeed what it represents. And then you can color the cells by whatever marker you want to look at. So here they're colored by CD45RA. And so again, the red cells here are the lowest for CD45RA, which makes sense. The memory cells are lower, and the naive and late stage effector cells are higher. And you can see this sort of gradient of CD45RA starting at the highest are these other pathway for some reason, and central memory cells also have high expression, and then they go down from there. So what I did to try to summarize this and relate it to a hypothesis that this principal component 2 represents a progression is that I gated, so I put these dots back into FlowJo and then gated on just the memory cells. And then I plot the average normalized intensity for each of the markers going from top to bottom here. So this will be the top, and then this will be the bottom. And so we're going from on this part, what looks like the most central memory-like, to the bottom, which looks like the most effector-like. And then you can see this phenotypic progression of surface markers here, and then also functional capacity. So you can see they lose the ability to make IL-2, and then gain the ability to make interferon gamma, followed by, MIP1 beta, and then CD107. And then at the very end here, we think this might represent what's talked about as exhaustion. Of course, in this case, it's stimulated with PMA and ionomycin, but perhaps these latest stage effector cells are progressively losing the capacity to do something. So it may be a more objective way to look at those populations if you incorporate the important parameters here. You can also look at the antigen-specific cells overlaid on top of this. And so again here, the naive cells are here, and then this spectrum of memory cells. And what I did here is I plotted the different antigen-specific cells. Yellow are flu-specific cells. These cyan cells are EBV-specific. And then the red are CMV-specific. And you can see this is just one example. There's diversity between people and between epitopes. But in this example, you can see that they sort of occupy different regions of this. And this is sort of representative of a lot of donors I've looked at where CMV-specific cells, more of them are further down on this differentiated branch, and EBV-specific cells are sort of all around, and flu-specific cells are mostly in the central memory compartment. I think this fits with what we would expect, because with flu, this person probably hasn't seen flu antigen recently, so those cells are just waiting for the next antigen, whereas these chronic infections, there is antigen there and driving the differentiation of these cells. Another kind of interesting aspect of this is this other swath of cells here, for which none of these viral-specific cells go into. So we think that maybe that other pathway represents cells specific for something else, maybe an intracellular bacteria or something like that. So we're working on that. But in terms of the analysis, as I've mentioned there's more to come here in terms of using nonlinear principal component analysis, and it's been suggested to use discriminant analysis to try to sort of parse out things that really define the differences between these cell subsets. For instance, the EBV and flu-specific cells are kind of overlapping here. Maybe another type of analysis would allow me to parse out better what these different populations represent. And I think one of the biggest challenges for this whole thing is how to deal with variability between antibody panels and day-to-day variation in signal and things like that. And so I think this is something we really need to work on, and how to standardize things, because it's hard to, once you start using these higher order analyses, it's going to be really hard to know how small differences in and technical things are going to translate into very large differences in the principal component analysis, et cetera. And so I think we need to think more about that. Also with rare populations, this principal component analysis really doesn't weight them into the picture much at all. It sort of tries to describe the variance across the entire population. And so rare populations that may be important, the differences that are in those populations aren't going to be well represented. And this is a case where this downsampling that they do with SPADE can really help. And possibly there are other ways of thinking about this for other ways of looking at it. And so, again, thank Mark, my boss, and Chen's lab, what we work closely with, Natalia, who's helped with a lot of the data that I showed, and also Bill, I took some of his data to show another example. And then again, with the CyTOF, it started as a collaboration with Sean in Gary's lab, and also talking to Aaron and other people in his lab. And then now we're using the CyTOF in Holden's lab together with Mike Leopold's help, who's done a great job maintaining that machine. And that's it. Thanks. Anything? All right. Brian?
So, you said that it's difficult to use SPADE to really compare across samples.
No, I mean, I said that SPADE, they actually have a pretty good way of comparing between samples because they load everything together.
Oh, right.
Whereas when I was doing the clustering analysis, or the simple approach, I don't have a simple way, I didn't develop any way of loading them together and showing two hierarchical clustergrams sort of that were loaded the same way, whereas SPADE is set up really nicely to do that. I think that's one of the biggest pros of the SPADE approach.
So I guess I'm wondering, could you replicate your PCA analysis using SPADE and reach a similar conclusion, or how common?
Probably, yeah. I mean, I've done that. I mean, I've looked at the SPADE analysis of what I had there, and what was fun is I actually loaded in my principal components into SPADE as well and plotted, okay, where are all the PC1 high cells in the SPADE? And it did make sense. Most of the big branches did correlate with like what principal components had, but there were some examples where there would be a branch, and then halfway down the branch it would shift in terms of a different component. So I don't really know. So like for instance, so the one that I was curious about were the cells that were lowest for my component 3, right? And that was actually kind of an end of one branch. It wasn't its own separate branch in the SPADE. I mean, you would have come to that in a similar way possibly, but who knows why they sort of look different, right? And so I think it's worthwhile to look at these in both ways and think about why it is we get sort of different visualizations. The thing I like about the PCA is just that you can see, I think it shows itself better. So I mean, it's not working well because it's all smeared together. So the smeared together maybe because of a technical thing. They're smearing together because we have noise, or they're smearing together because PCA is not good at discriminating them. But at least you can see that they're smearing together, right? So at least the minimal thing with PCA is that you can sort of get an intuitive feeling for how discrete your populations are by this analysis. So I think, whereas with the clustering analysis, you need to then go and ask, okay, are these really different, and how are they different?
Right.
Once they're clustered, they look different. Yeah, they look different, and that may be true, or that may not be true. And here, it looks like they're all kind of a big smear, and that may be true, or it may not be true. But at least you're sort of underplaying it here. So when they're separate, they're probably very separate. That's the hope, maybe, but who knows? So we need to still talk to people that know what they're doing to figure this all out.